All right, now I am live. Hello, everybody. Welcome to another video blog from Monarch Science. I'm Andy Davis, the author of this blog site, and once again, I'm coming to you from my messy office here at the University of Georgia. If you are watching this, then that means you are a fan of monarch butterflies and also the science and the study of monarchs. At this blog site, I do my best to break down the latest studies of monarchs for folks who aren't science geeks like myself. And today I'm doing a video blog session, obviously, and I have a very special guest, Dr. Elise Zipkin from Michigan State University. Dr. Zipkin is no stranger to the world of monarch research. She is what I would call a quantitative ecologist, which I think for people at home means someone who studies the natural world, but like using massive amounts of data and very sophisticated statistics. So that's another way of saying that she's really, really smart. In fact, most of her analytical approaches are well over my own head. So I'm talking with Elise today because her lab just published a brand new study looking at how climate change affects monarchs. It was spearheaded by a postdoc in the Zipkin lab, Dr. Erin Zylstra, I hope I said that right, who couldn't be here today. And I'm hoping that today we can chat about this paper and help folks understand what the team did and what these results mean for monarchs. So welcome, Elise.
Thank you. Thanks for having me.
Sure. I'm glad to have you on my blog. We've known each other for many years, and I've always been a big fan of your lab's work.
Oh, thank you.
For people at home, let me share my screen and I can point people to the direction of your lab webpage here, just to show everybody all the crazy things that you do. I mean, your lab is so productive. How can you be so productive?
Well, I'll tell you one thing is I work with amazing people. I mean, Erin, who is the lead author on this new monarch study, but also like all the other students and postdocs in the lab, they're just super fantastic. They're really, exciting people that are interested in really cool questions. And also, I think we have really good collaborators. Like on this project, that we're talking about today. I mean, Leslie Reese has been a long time collaborator of mine, and I love working with her. And our other collaborators down in Mexico. I mean, we just have a lot of people that we really enjoy working with, and I think that together helps the productivity. And honestly, I think one thing that's kind of interesting about quantitative ecology is, in some ways our questions are always really simple. For the monarchs, it's like, why are monarchs declining? And this, you have this picture here with the snake from Science, and that was just really what happens when you lose amphibians? What happens to snakes when you lose amphibians? And so our questions tend to be kind of simple, but what it is, I like to say, is we try to answer these questions that no one else knows how to do. So we're constantly making lemonade out of lemons. How do we figure out the question is simple, but oftentimes the data are messy or incomplete, and it's hard to answer it for a variety of different reasons, maybe something about the life history of the species. And so we're trying to put together all those data and statistical and mathematical models and figure out a robust answer to these kinds of questions.
So I'm going to click your publications section here just to brag on you a little bit more. You got 4 in press. I guess this one's not in press anymore, but this is a pretty substantial publications page, and kudos to your lab for this. I mean, you've got some great people, but you're obviously leading the charge here. Mm-hmm. This one here that you did in where was the snake one?
The snake one, 2020, a little bit.
Oh, yeah.
So the next.
And you're a first author in that one?
Yeah.
How can you go from snakes to monarchs?
Right. So that's a good question. All right. So I hate to break it to the people who love monarchs and butterflies, but there are a lot more similarities between things that are alive than there are differences. So in a lot of ways, we can use similar methods, to think about problems across different taxonomic groups. However, the caveat I will say for that is that we always work extremely closely with people who are experts on those taxonomic groups. So for example, that snake paper, my collaborator Karen Lips, she's the one who led the data collection of these amphibians and snakes over, more than a decade. in Panama. So we always work, with other collaborators who really know about the taxonomic group, so they can rein us in when we're like, okay, we think this model makes sense because here's our assumption about what this species does, and they're like, no, I know your butterflies do that, but snakes don't do that. So they do kind of reel us back in, but we lose, a lot of kind of similar kinds of statistical properties to think about things. A big thing that we think about in this hierarchical modeling framework, which is a lot we do, is we think about modeling the biological process, and on top of that, modeling the sampling process. So any biases that we see from how data are collected. And that kind of, runs universally true across wildlife systems and how we collect data on wildlife.
Awesome. I like it. Why don't we segue into the into this new paper? I'll share the screen here if you want, or do you want to share your screen? Or
no, I don't have anything up. You can share this,
okay.
So then you can direct questions.
Let me share the screen here. I've got your paper pulled up here, and let's just look at the first page here, just so we can show everybody what we're talking about. So you got this paper accepted and print and published in Nature, Ecology, and Evolution. Congrats on that. That is a stellar accomplishment. I mean, Erin should take a lot of credit for this, right?
Oh yeah, she did all the hard lifting. I'm just in the background, like, oh, have you tried this? Have you thought about that?
and I see you've got a lot of other sort of well-known names in the monarch world. You got Karen O, you got Eduardo from Mexico. Sarah Saunders, does she still work in your lab, or is she?
So she's a former postdoc in my lab who now works for Audubon Society. And this part of the work, is something that she started the ideas for while she was still a postdoc in my lab. And she, continued to kind of help and guide and obviously provide really great insights on it.
So I've read the paper a couple of times, and I've seen some of the pressers that have come out on this. And maybe I can just sort of throw you some of the first questions here. And these are just softballs. Maybe you can just tell me what's the spiel that you usually give to the to the journalists about this project, like why you did it, what you found, and what do you think it means?
Yeah. So, I've been working on the monarch world, as you know, for a while, so more than a decade now. And really, Leslie Reese, a collaborator on this, was the one who kind of brought me into studying monarchs, right? We were in a working group, I think, more like probably close to 15 years ago, something like that.
Is that the one was that with me, you mean?
Yeah, with you, remember?
That was your first foray into the world of monarchs?
Yes.
Oh.
That working group is what brought me into working on monarchs. So that was like 2009. I guess it wasn't 15 years ago, 2009. Yeah.
Those were some fun times.
Yeah, that was really fun. So, we started out kind of looking at different climate and land use effects of monarchs. And really we were focused primarily on the breeding season and the Midwest part, and a little bit on the effects from what's happening in climate in Texas. But, in our back of our mind, Leslie and I were always talking about, and Karen too, this is something we thought about a long time ago, which is we really wanted to think about this question of, can we quantify the relative contribution of all the stressors up to monarchs along the migratory route to look at why monarchs are declining? Because there's some really, really great work out there on, all these different parts of the migratory cycle where, we had shown stuff like, here's how weather is impacting monarchs and the breeding period. Here's how glyphosate, people had done a lot of experimental work with glyphosate and looking at how, that had applications of glyphosate had led to lower levels of milkweed and how that has led to lower levels of monarchs. There was also really some important work on looking at fall migration, some of which we had done, Sarah had led, but also in other places showing the disconnect between apparent disconnect between the summer population size and the winter population size. So there was a lot of different research around the time, but we really wanted to bring it together. And the other kinds, we're not the first people that thought of this, and other people had brought this together, but generally they weren't bringing the different data sources together. So people were doing more like theoretical models through the cycle to try and pinpoint, what was important, or they were only using the winter data and including other covariates. So we thought it was important to include as much monarch data as we could. So we used 6 different.
Let me scroll to the part of the paper where you're going to get to that. It's down here right here, right?
Yes, so that's the locations of the surveys. So from two different time periods. So I guess what I'll say for this is our original goal was we really wanted to go back to when systematic data started being collected on monarch in the overwintering grounds. So the mid 1990s, and estimate the full, what had happened from the mid 1990s all the way up to 2018, which is the last year we had the data. But what we found is, like, pretty quickly as we started doing our analysis, aggregating all the data across the Midwest, aggregating the data from the wintering colonies, the data just really weren't available during that early time period, which is the mid 1990s to the mid 2000s, to look at why, to really be looking at the relative contributions of all those stressors along the migratory route. So there was just a lot less data locations, a lot less surveys that had been done, these systematic surveys. And unfortunately, although we really wanted to kind of look at the relative contribution and structural decline during that time, which was the steepest monarch decline and the time period when glyphosate application rates really increased, we were just really not able to do that so well. So we focused primarily on the time period from 2004 to 2018, so a 15-year period. And one thing that's kind of interesting about that time period is the the glyphosate application rates has kind of leveled off. So that's like figure one. What we had done, what we'd found, there'd been some kind of, people thought this, but actually what we did, I mean, this was one of the things that took a really long time, figure 1B there, which is just the county level amount of glyphosate application that had been sprayed across the Midwest. And that took a long time to collect those data and calculate these values. We had done it, probably, Andy, you'd seen, we'd done it before for Illinois, but Aaron did this across the entire Midwest. And so from 2004 onward, maybe a little bit afterwards, that amount of glyphosate application rate kind of levels off. And you can also see, if you look at the overwintering population, there is less of a decline during that time period. So the steeper decline kind of happens in that first decade from when we have data available, and then less so in this more recent time periods. So the glyphosate application rates were much more stable. There was spatial variation. There was some, it did vary from year to year, but it was much more stable. So we focused on this time period. We put together all the data. There were about more than 14,000 surveys across the Midwest during this time. And yep, this is this one here. And we put it together, and then we did this thing called hierarchical partitioning, which is essentially where we exclude and include variables of our model, different types of variables. So you could go up to figure 2 here in the paper, and this will show you. So we did this by groups, because our question wasn't how was each specific variable influencing our influencing monarchs, because there's a lot of variables and interactions among variables in our model, but we broke it down by these categories, which, so if you look at figure 2D here, so that's the bottom left corner there, it's like, what's the relative contribution of these different categories? The size of the late wintering population, the spring weather conditions in Texas, the breeding areas in Texas, the summer weather conditions across the Midwest, and then the summer crop cover and herbicide. What's the relative contribution of each of these 4 different groups of variables to the size of the monarch population, the peak size of the monarch population, basically?
And so that's derived from the counts of monarchs, of adult monarchs in the summer in the Midwest.
It's not, yes, it's not exactly derived. It is modeled by that. So that peak size, unlike in previous times where we've used, the general way that people have looked at the monarch population size in the summer is they restrict over to a certain time period, what they call peak summer, which is, around the August time period in the Midwest. And then they look at these NABA surveys. So those are North American Butterfly Association surveys across the Midwest. And they look at what, an index of what the size average, maybe of what the size of the population looks like. So instead, what we did is we modeled this across the counties in the Midwest. So that's like all those areas in the red, like the orangey red color in B up there.
Okay.
we modeled during that peak time, what was the size of the population in each of those counties? And essentially we take an average of that to get what is the index for the population. So yeah.
Yeah, just to slow you down a little bit, when you say you modeled the abundance of monarchs, is that another way to say you extrapolated in from areas where you do have surveys to the areas where you don't have surveys?
So yes, we did do that. We modeled it with all the covariates to estimate what was the size of the population in each of the counties. And we did do what you're saying, where in counties where we didn't have monarch surveys, because for sure we don't have data from all those counties, we estimated the what would be the county level size of the population. And then we took an average of that. Now, I'll do say though, because one of the things that we're really keen on, like, you know, as a quantitative ecologist, as you know, Andy, you can always make a lot of different decisions about what makes sense or doesn't make sense in a model. And the best way to figure out if what you're doing is good is to see how robust those results are to many different kinds of decisions that are all logical. Right? So we did this a lot of different ways to figure out if what we were doing was problematic. So for example, we didn't include the counties that we had data and we looked at an index there. We changed the window dates of this slightly. We looked at, all these different indexes away, and we got all values that were highly correlated. So none of these decisions that we were making on exactly how to estimate the peak summer population size had a huge difference in what the index was. And that's also because it was an average over that whole region, right? So if you look down, I think there's an appendix or there's a figure in the appendix that shows like what is that peak population size. Yeah, so that would be that figure here. And then you can see, unlike, you know, the winter area where for sure, you know, we have only an estimate of the colony size, but we have no error bars around that estimate. And that is one tricky thing about, you know, looking at the winter colony data. But here's an estimate of what we think those summer population sizes were, the peak summer population size. You know, as this index is represented as the amount of monarchs that you would count adults per party hour, basically in this NABA survey. That's like, it's standardized this way. And you can see these errors around that based on those data from the summer. So
and these end in 2018, so that's the last day.
Right, that us here. These values are very highly correlated with what the winter values are subsequently, but you should know that no data from the winter, right, influences how we estimate these counts, right? So, and we do still find the decline, although the decline is not quite as severe. And also, I think this does do a good job of showing like some of these years early on where there was high, you know, values, where there were these high counts. I mean, we do have a lot of uncertainty on some of those, right? So,
all right, so let me see if I can paraphrase, and you can stop me if I'm wrong. So the gist of the study was that you had data from the Midwest on monarch abundance, and you wanted to know what factors are causing the monarchs to be abundant one year or low another year, and et cetera. And you wanted to see if there's any factors that are causing a long-term trend in the abundance of monarchs in this area right here. And so
yeah, so all in there, right? And it's not just through time, it's also through space. So we were able to do that, right? Like we take in those spatial variations too, which could have a big effect, right? Like the amount of crop area locally and how much glyphosate was sprayed. So you get this estimate. I don't think we have too much in there. There might be some figures. There are some figures in the appendix, I think, but we didn't show this, but you know, we do have a spatial understanding of how the population also changed its size from year to year.
So that means if you have like one county over here that has a lot of monarchs, you wanted to know why that was compared to another county over here that didn't have very many monarchs.
Right. So we tried to use these covariate values to explain that.
All right. So then you factored in, you know, whether the late winter population size affects the breeding size, whether the spring weather here in Texas affects the the summer population size, whether the summer weather and whether the amount of crops and whether the amount of glyphosate used or Roundup used also affected. And down here is where you have your big kahuna result, right?
Yeah, so on D, you can see how much each of these variables influence the size of the summer population. And so what you see here is like spring weather and summer weather combined were explaining most of the variation in the size of the summer population.
From year to year, right? The year-to-year variation.
I mean, and also spatially too.
Spatial.
Mostly year, I mean, yes, you're right. The bigger thing to think about really is the year to year, but also that spatial variation does come in with it. Although you're right, because that's more, the spatial variation is really more summer weather because we don't have a spatial component to the spring part. So I do want to make one interesting point too, which I think like might interest some of your listeners. You know, what we had originally had to do, so you can see what's missing is that we don't have spring monarch data, right, included in here. And we had originally wanted to include that. And we thought about different ways that we might do that. But it turns out that we wanted to use systematic data. So that's like, we thought about using kind of Journey North data or other kinds of data where there's opportunistic observations. And by the way, those data are extremely valuable. And I can tell you a little bit about how we're using some of these opportunistic data in future analyses that my grad student, Matt Farr, is working on. So I think those data are really valuable. But for this analysis, when we were trying to look at abundance, not having information about where things aren't tends to be really, really problematic. And so, you know, we thought about, there are some systematic surveys in Texas that are similar to the state surveys that happen up in the Midwest, but they're really much shorter time period. So there's fewer sites and it hasn't been going on quite as long. And then the other thing I wanted to say, that just kind of a side caveat, after Hurricane Harvey, Leslie and I had started out trying to do some systematic surveying of monarchs to figure out like if there was effects of Harvey. We'd gotten this rapid NSF grant. And, you know, we worked with some people. Neither one of us did the data collection, although Leslie might have been out for a little bit. Anyways, what we found when we put out these systematic transects is we got almost entirely zeros. Almost all monarchs have zeros on these transects. And I think it has a lot to do that with in Texas, you're talking about a huge area that the monarchs occur on. And the size of the population is essentially the smallest that it is at any time of the annual cycle. So it becomes really, really hard to systematically survey for monarchs. And, you know, this is kind of an interesting thing because this came up, you know, with the snake thing, but it's like, there's this idea that you just go out and collect more data, but there's not going to be like a good way to collect systematic data of monarchs in the spring because they really occur in a very low abundance over a very large range. So that was something that we kind of figured out through this process too, because it would be great. And actually, like I was saying, my grad student, Matt, is working on this summer model of how we can include all these opportunistic data to look at spatially where monarchs are across the landscape and how that might be changing from year to year, which I think is really important because we're clearly seeing that spring, and not our analyses, but others too, that spring is a very key and important time for how well monarchs do each year. But yeah, so yeah, that was kind of a long digression, but I think it's kind of important.
Well, let's get to the next part here. So you modeled, you found that the climate really matters for the summer monarch abundance. And what was it that mattered? Was it wet or hot or what?
Right. Okay. So in the spring part, so the spring data is not really spatial data. That's like kind of an aggregate across eastern Texas. And so for spring weather, what we found is that monarchs in the summer tended to do best when spring weather was near its average values. So about average precipitation, average temperature, maybe a little bit warmer, cumulative temperature, which we measure in growing degree day. But about averages were the best. In summer, this was really interesting nuanced results. So you could go to figure, I think it's figure 5, maybe, which is the summer weather one. Because I think that one is really, this is the spring weather, so that's about the average stuff, but the summer weather one is really interesting. So this I think is really interesting. So what we found is that, let's go into the top right corner. Yep, where you are.
Here, yep.
there's a warm, average, and cool location. So essentially you can really think about that in the Midwest, that's really latitude variable. So the warmest ones are Ohio, you know, then mid, and then the highest ones are up Minnesota or Michigan, those kind of areas. So what we found in those cool areas is that if you were hotter than average, there were more monarchs, okay, available. However, in the lower level latitudes, so in the warmer areas, as it got too hot, actually the expected count, you know, per locations went down. So the size went down.
So to get more monarchs, we just need our cool latitude areas to be warmer.
Yes, but let me show you what's happening on the top left. And I thought this was really interesting. So this is the difference in the average GDP between 1994 to 2003, and the difference from 1993 2003 from the second time period 2004 to 2018. Man, I see that that's labeled a little bit. Dang, it should be right in the
I hate that too when you find.
I know, I never read them afterwards. I'm sure it's right in the legend because we read that over and over. So this is the change in the difference. So if you're at 0, that meant that a site, so like where the y-axis is, if you're on 0, that meant your site was the same, had the average basically temperature in the second time period than in the first time period, okay?
Okay. Yep.
If you're above that site, you got warmer. If you were below that site, you got cooler. So, and this is also confusing, but this is what this average GDD, right? Oh, no, that is right. Average GDD on the top, on the scale. That's labeled right. So this is what the average GDD was in 1994 to 2003. So if you're 500, you're a cool site, right? If you're 1100, you're a hot site. So those hotter sites on the, you know, the further to the right are the southern latitude, and further to the left are the higher latitude. So essentially then what this is saying is those ones that are further to the left, right? So these are the different dashed lines, like that solid line, a cool site. A lot of those sites became cooler. They didn't get warmer. So you're in like the worst situation. They're cool. They actually became cooler, which would be less monarchs. And a lot of those sites that were hotter, so you're on the right side of the graph right there, those sites became hotter. So you're in kind of the worst situation in the south and the north.
When you say south and north, you mean you don't mean southern US, you mean southern portion of the Midwest.
Of the summer, right, exactly, of the summer breeding range. So those areas in Ohio or whatever got hotter, and the areas further north got a little bit cooler on average. Now, that's not to say that will continue to happen. Those aren't actually the projections for climate change into the future. That's just what happened between that first decade and that second 15-year time period. It just that's what we found happened, which I thought was really, really interesting. So it kind of happened that 15 years was like in the Midwest was like not the greatest, you know, climate. The hotter places were actually a little bit hotter, which is worse for monarchs. And the cooler places, which tend to do better if things are hotter, were a little bit cooler on average.
All right, so we're getting there. So you found that the abundance of monarchs in the summer is really affected by the climate in the summer, and also especially in the spring. And if things aren't, if the temperatures are off from the average, that's when you start to see declines in the monarch breeding.
If the temperatures in the spring are off the average, that's when we see decline in monarch. In the summer breeding ground, lower latitudes that were hotter led to less monarchs, whereas like in the higher latitudes, being warmer than average did lead to more monarchs.
All right, I got that part.
Latitudinal gradient.
So now you also did the same thing going backwards, looking at the fall migration. You wanted to see if the summer population size affected the ultimate colony size in the winter, right? And then you also included the autumn nectar resources and the forest cover. This is figure E.
Right. So then we did the same thing with groups of variables there, which is that same hierarchical partitioning, and looked at, you know, including groups of variables in and out, and how much did that influence it. And what we found is, you know, about 90% of the variation from year to year in the winter population size was attributable to the size of the summer population. And that's not to say that the other variables didn't have an effect, but they were smaller. So I do want to make one note, which I think is important. I don't think our results turn over across the summer or the winter, turn over anything that's been shown before. Like, we do show that when there's higher levels of crop and higher levels of glyphosate, like, there were less monarchs. I mean, there is an effect that way. It's the same thing with autumn nectar and early winter forest colony cover. When we see, more higher quality dense forest cover, we see larger colony sizes there. So all of our covariates were going in those same directions of what we expected and what had been shown in previous research by us and many, many others. It's just that what the big thing is that we were trying to figure out what was the relative contribution of all of those things to the size of that population.
To the year-to-year variation in the size, right?
Yes.
So I got that part.
It definitely, well, so that's both year to year in the winter size, but it's also, there's the spatial variation in the colony. So we're modeling each of the individual colonies in the overwintering sites. So it does account for that spatial variation, right?
So what I don't get though is, so I know you're saying that the abundance of monarchs in the summer is correlated with the size of the winter colonies, and the abundance of monarchs in the summer is dependent on the climate from year to year. So what then is driving the decline in the winter colonies?
Right. So unfortunately, with the hierarchical partitioning, what we can't do is put this all together with the size of the population. But our conclusion, based from the fact that the summer population size was explaining more than 90% of the variation, and that the summer of the winter population size, and that the winter and that the summer population size had been largely explained by the spring and summer climate weather variables, is that the spring and summer weather variables are the largest contributors to explaining the size of the peaks, the size of the monarch population each year.
The size of the breeding population.
Well, and the wintering population, because what we see in
because they're correlated.
Well, because of basically that big column with the butterfly in E.
So as long as you're saying, as long as you get a really good recruitment each year of monarchs, then you tend to get a larger winter colony size.
Right.
So then
it has a strong positive correlation. So if you look at figure, it might be the next figure. It's either figure 3 or figure 4. I think it's figure 3. Oh no, is that figure 3? Figure 4. Go to figure 4. Okay, go to figure 5. This one. This is the correlations between each, the size of the populations at three different points. So here's where we have monarch data. We have monarch data essentially that we had good monarch data 3 times the year on the size. We have the early winter population size. We have an aggregate of what the size is at the end of the winter. This is a little bit more trickier data. We only had one point. We don't know them at each of the colonies, but overall. And then we have this peak population size of the summer size that was based on our model-based derivatives. So this is a model-based values. The ones without error bars, right, are because those are data collected in the wintering colonies, and the one with error bars are our data collected as an estimate, or a model-based estimate of the population size. So what we did, these are just post hoc, essentially like correlations, regressions, where we're just like looking at the size of the population between each of the different time periods. So if you look at the top, it's like, what's the relationship between the summer population to the early winter population, okay? So we find like this pretty high R squared, 0.67. So there's like a pretty high R squared, taking into the uncertainty that we have about what is the summer population size. Then if you look at what's the early winter to late winter, still like a pretty high one, 0.4, the R squared is 0.4. So like a little bit lower. So that's like mortality over the time plane of the winter. And then the next one, the bottom one, is that's late winter population size to the subsequent summer. And that's the lowest correlation. We only find the 0.3. And that's because we argue, right, that these other kinds of variables are having the biggest effect that time. So this is just providing further evidence that the time frame when we have the lowest correlations on these things is that time between what's the size of the late winter population and how big the peak summer population gets.
So I totally get that they're correlated. And in any given year, if you have a large summer population, you can get a large winter population. So I'm still questioning though, does that mean, so is what you're saying here that the cause of the decline in Mexico is because there's just not enough monarchs being produced in the summer?
I mean, that's what we're saying for the last like 15 years. And so I think what you're trying to get at is this question of like, how are you describing year-to-year variation versus the like decline in the population? Is that what you're?
I want to get to the trend. Like I know that
Right.
year to year is correlated, yeah, but the trend downward in Mexico is what I'm after.
Right. So so the thing about it is this is just like, I mean, it's an excellent question. It's something reviewers bring up. It's something we thought a lot about. It's something that everybody thinks about. How do you know you're explaining, we get that you're explaining year-to-year variation. How do you know you're actually describing well the decline, right? And so the one thing that I think is worth putting out, right, is that they're not separate. Sure, you can do these kind of detrend sort of things, but there's a lot of assumptions associated with that. And so our approach is like, we want to do it more holistically, but we really wanted to get at that answer, right? Like, does our model do well at describing the decline and not just the variation? So if you go all the way down, and I'm going to forget what number this is one of the extended data figures where we tried to look at the residuals over the time period of 2004, 2018.
Oh, I think I remember seeing this.
This one
here? Yeah.
Yes, I think this is a good one. Okay, so a couple things I want to comment about that is important to note. It's unfortunate based on our analysis. Like I said, from 2004 to 2018, the population in the winter data has declined a lot less than the first time period. Now, it does depend on what you choose as the beginning and the end of the start date, but you know, we have these these reasons related to the data, basically that we chose that 2004 date, right? If you chose different start and end points, right, that can lead to a different decline, but the decline from 2004 to 2018, especially since 2004 was a really low year and 2018 was a high year, is much more modest, right? And I think that that is important to note related to the decline. But what we did here is we wanted to look at, all right, how well is our model describing the year-to-year variation and the decline? So what we did is a couple of different things. The first is we looked at the residuals of our full model, and we looked at residuals of our model where we pulled out those summer and spring weather variables. Okay, what if we don't have them in there? So two things. This figure here shows the residuals for the full model. And what you find is there's no trend in the residuals. So that's 0. I don't think we have this here, but I know it's somewhere discussed in the paper. If you have a model without the spring and summer variables, you do still find a little bit of, there's a slight decline in the residuals. So the thing is, a model without the spring and summer variables doesn't describe the decline completely. So we know that from the residuals. So the other thing here is that I think is really important. What you can see is with these different variables, how well do we describe what's going on? So in each year, right, like, are we explaining the fluctuations and the trend well? So our model does describe the trend, although the trend is less severe than in other time periods. But also this is, I think, a year to year, which is kind of interesting. Okay, so in a lot of times, so what we find is each of these points are like each colony. This is the trend of each, or this is the residual of each colony and how well. So for example, like the first year, 2004, that's really well, right? Because it's like the middle point is right on that line of 0, and there's some points above and some points below, right? And so there's these different residuals where sometimes it looks really good, it's right centered on there, and sometimes not. And so like what we want to see is like kind of each year averaged around 0 and some above and some below. But what's interesting is I think there's like 3 years that are kind of interesting. That's 2012, 13, and 14. If you look in there, that's when our model doesn't actually describe those years quite as well. And those are those are like three very low years. So our model does slightly overestimate what the size of the population is in those 3 years that were really, really low. So yeah, I mean, I don't think that our model describes everything. We clearly couldn't there were reasons why some things we weren't able to include in there. But for the most part, it describes pretty well kind of that trend and the fluctuations. But there are some years, and those are the lowest years, where I'd say our model does poorer than in other times.
All right. Well, I think I got most of that, you know. So to be fair, you know, a lot of this is over my head as well. And maybe to some of our viewers at home, I think can we say that the take-home points of your study is that climate really matters for the abundance of monarchs, both in the summer and in the winter?
Right, the size of, right, spring and summer climate conditions model quite a bit for how big the population size is at the end of the summer, and the end of the summer population size matters quite a bit for how big the size of the winter population is.
All right, so that's a good segue into maybe my last question for you, and this is probably the hard question I've got for you. Looking at this figure here, figure D, this is maybe it's a philosophical question for you. If it looks here like the late winter population size does not really factor into the size of the summer. So does it matter if the winter population size is big or small? I guess is the question.
I mean, it's a great question, right? I mean, so that's a really good question. I mean, I don't have a great answer for that. I would say a couple things. My guess, and this is speculative, I would say my guess is you need it probably above some threshold size, right, to be able to like regenerate, you know, in some ways, but that how much that kind of regeneration happens really relates more to abiotic features than the size there. I mean, it doesn't, in some ways it doesn't surprise me because, you know, insects, we know a lot about insects, that they're really affected by weather, their growth, their survival, all sorts of things related to that. And if you think about that, right, the late winter population size is essentially the smallest that it is all throughout the annual cycle, right? Because they've only just been dying since the end of the summer at that point. So the size of the population is pretty small. So there's not going to be that huge, I mean, there is sometimes a really big one, but a lot of times there's not like huge variation. And my guess is if that population is small versus not quite as small, that might not be the biggest thing that influences like well, we know it isn't the biggest thing of what influences the monarch population.
So then why should we track the numbers in Mexico if they don't really matter statistically speaking?
Well, first of all, I think that what happens in the early winter does matter, right? Like what's the size of the population, because that is our best metric for understanding long-term what the kind of essentially, I would say an annual metric, either at the end of the summer or at the beginning of the winter is a good metric of, you know, what the size of the population is. But I think I agree with you that the end of the winter size of that population is not a particularly meaningful number. Now, it might also be, you know, I know there's variation from time to time and when they leave. So they're always trying to do this kind of at a specific time. You know, I think we took like late February counts, which I think is one of the last times that they're all there before they start migrating. But there's obviously some sort of variation in that. But I do agree with you that that size of the population at the end of the winter is not maybe that helpful in understanding sort of
how well they're going to do the next year.
Yeah, and also it wouldn't be the metric I would choose for look at how well the population has done through time. You know what I mean? Because that value sort of isn't a good indicator of that. But I think there is something to be said for the fact that we find sort of this consistent story in this early winter population, which really is a time frame, you know, when the monarch population is all there, and those indices of the population size at the end of the summer as well, right? There's a high correlation in that I wouldn't expect that correlation to be 100% because there is mortality over the winter. We do, or sorry, over the fall migration and when they resettle, and that those do matter based on covariate effects, like what those conditions are like in each year. But, you know, something I think is causing kind of these longer term trends that we are seeing. And I think that the answer is a lot, right? It's these external abiotic factors, and most recently, maybe weather.
I forgot how fun it was to talk to you about your research.
Oh, thank you. That's so
I mean, I always learn something talking to you, because this is, even though I study monarchs, I mean, I don't do this kind of work. I mean, I think I learned something today.
Oh, I learned so much too. Somebody was just emailing me about predators on caterpillars, like larger caterpillars. And I was like, I don't know. And I roped in Doug Landis. I don't know if you know who he is. He's at Michigan State, and he's been doing all these interesting survival things. And he was telling me like all about this and the predation, you know, on these egg and instar stages. And it was so interesting to me because I was like, man, I never think about that. I always think about this like really big picture, like full annual cycle stuff. And I was like, I should read more about that and think about that. So I think that happens to all of us. Yeah.
Well, you know, Elise, I really enjoy your enthusiasm and your energy. So I think we should probably wrap it up, because I know you got your next meeting here. I want to thank you for taking the time to indulge me in this blog, and for telling everybody what your study found. You know, I look forward to seeing much more from your lab. You know, if you've read my blog, I plug your stuff a lot. So
I've seen it in the past. Yeah, I really appreciate that. Yeah.
All right. Well, I'm going to let you go. And
thanks for having me.
Thanks for the viewers for tuning in. And we'll look forward to more from Elise's lab in the future. All right. See you later.